Ooh. Okay, here we go on a voyage into Krebs Cycle. So, Krebs Cycle, what goes into Krebs Cycle? Now, if you remember, at the end of the link reaction, we have made this thing called acetyl coenzyme A. And this coenzyme A is just a little helper molecule. And so really the first thing that happens is once it's sort of helped the acetyl 2-carbon fragment go into Krebs cycle, it's leaving. So it's kind of a bit like a Boy Scout. It helps the old lady across the road, then it goes back to collect another one. So the first thing that happens is that coenzyme A, having just attached itself to the acetate portion, leaves to go and collect another one. So what happens to the acetate portion, that 2-carbon fragment, is it gets added or joined, so this is a joining reaction, to a 4-carbon compound. Now, you don't need to know its name, it's got a name, it's a lovely name. It's called oxaloacetic acid. Love it. One of my favourite flavours. And when you join a 2-carbon to a 4-carbon compound, it will not surprise you in the least to know that you get a 6-carbon compound. And again, this has a lovely name. I'm going to call it citrate. I'm going to name the other one. And really what Krebs cycle is about, Krebs, absolute genius, worked out that these reactions could go around in circles. So what we're going to do is we're going to make 6-carbon compound back into the 4-carbon compound. Again, series of enzyme controlled reactions. So obviously if we're going to make a 6-carbon compound into a 4-carbon compound, we're going to go through a 5-carbon compound. And to get from 6 to 5 carbons, we're going to need to remove a carbon dioxide. So again, this enzyme that removes it is the decarboxylase. And we're also going to oxidize our citrate. So that means we're going to remove a hydrogen and take the hydrogen off. Give it to NAD, that coenzyme, to carry it about and make an NADH. And because we've taken 1 carbon away from our 6-carbon compound, we get left with a 5-carbon compound, which also has a name. We don't care about it. And then I think you can see where we're going with this. We're going to make this 5-carbon compound into a 4-carbon compound. So again, to make 5 carbons into 4 carbons, what are we going to do? We're going to take off the carbon dioxide. We're going to use decarboxylase. I'm hoping I've left enough room there. Oh yeah. Oh, look at me with my spatial awareness. And incidentally, again, we're going to make, in that process of removing stuff, we're going to make an ATP. And again, this is not involving chemiosmosis, it's not involving the electron transport chain, and this is again substrate level phosphorylation. Why phosphorylation? Well, if we put the other end of the reaction on, we're going to phosphorylate an ADP. And again, this 5-carbon intermediate is going to be dehydrogenated. Remove a hydrogen, pass it to NAD, make NADH, and we're also going to take off yet another hydrogen, and this time we're going to pass it to FAD, that different coenzyme. and that'll make FADH. Now you'll see these reduced coenzymes written a number of ways: FADH plus H plus, NADH2. Don't worry about the numbers, that's what I'm going to say to you. And between the 4-carbon compound and where it can join on to the acetate fragment, we're going to remove yet another hydrogen. Again, we're going to pass that to NAD and make NADH. So you kind of need to learn that, and in terms of exam questions, so you can see it's a cycle. In terms of exam questions, they may ask you to, they might leave some blanks and say, you know, hey, what does this letter Y or Z represent? And you should be able to work that out. They might give you some of the additional intermediates. So this is a hugely simplistic Krebs cycle. There are loads of intermediates with really jolly names: succinate and malate and fumarate and all sorts of things. And they might ask you to work out where the carbon's removed by giving you the number of carbons of each of those intermediates. So there are things to look out for. The other sort of direction they might go is the things that can go wrong. So, you know, a very good way of testing your understanding of a series of reactions is to ask you about things that go wrong. So they might ask you something like, you know, why might you get a build-up of this? You know, well, okay, that bit of the reaction's going okay, but it's not being converted because one of these enzymes isn't working to remove the carbon dioxide or hydrogen. Of course, eventually that means you'll run out of the 4-carbon compound and the whole thing will come to a great big grinding halt. So that might be one way that they ask about it. And some of the mitochondrial disorders certainly do that kind of thing. There's sort of, some of the enzymes are not working in that and they're causing build-up of one or other compound. And they might ask you then the impact of that on the electron transport chain, which is of course where all of these reduced coenzymes, they all end up in that electron transfer chain. And if you need to go back over that, the sort of the overview is the one that talks about the NADH all going to the electron transport chain. So, get learning.